Well, we continue with 1212b here. Just briefly got started with the Surmounting the Cartesian Cut, Klein Bottle Logophysics by Diego Lucio Rappaport. The DA, which is the direct algebra, and its double tetrahedron codifications in NUCRS, is found to be associated with a pair of superposed Klein bottles, yielding the double 3D space and momentum space of physics, and the double helix of DNA, which thus appears related to a pair of Klein bottles, being the case that only one is necessary to construct the genome. We show that the depth-time variable of visual perception materializes as space. Thus, the DA is derivable from two Klein bottles, and the purported dualism of physics as presented in NUCRS is rendered to be based in the non-dualistic Klein bottle logic. We discussed the relations with the theory of evolutions and between biological periodicities and the Klein bottle of the Mendeleev table. We present relations between metacognition and the hyperklein bottle and evolution in terms of time waves defining in toto the different structures. We present rudiments of the ontoepistemology of the hyperklein bottle, and evolution in terms of time waves defining in toto the different structures. We present rudiments of the ontoepistemology of the hyperklein bottle logic, its relations to the anthropic principles and the universal physical constants, which are found to be contextuals. Introduction. A logophysical theory surmounting the conception that separates the objective and the subjective realms. Cartesian cut, based upon Klein bottle logic. And torsion fields was proposed and applied to cell biology and embryology in the accompanying article, Rappaport 2011. The objective of this article is to extend this conception to genetics, its relations to quantum mechanics, and a fortiori with embryological development and evolution, and to show that the origin of and the double 3D space coordinates and the momentum representation of physics and of the dualism at large claimed by theoretical physics stems instead of the purported dualistic basis from the non-dual Klein bottle logic. With respect to the genome, we shall present several codifications of genetic code GC, and the self-referential topologies of it. To further apply it embryological differentiation and to relate it with a nilpotent universal computational rewrite system, the NUCRS by Hill and Rowlands. This theory is important since it produces from a self-referential syntax much of mathematics and physics fundamental symmetries in terms of a codification of them that shows that NUCRS has for its mathematical backbone the Dirac algebra of quantum mechanics. In doing this, it shows how space, momentum, and the fundamental interactions, holography, etc., are all derived from self-referential syntax. We shall present a simple derivation of the genetic code from the Klein-Bottle logic to further related the fundamental 64 codons to the Dirac algebra of quantum mechanics. In particular, we shall show that the origin of the double helix and the double 3D space coordinates and momentum representation of physics and of the dualism at large claimed by theoretical physics and biology stems instead of the purported dualistic basis from the non-dual Klein-Bottle logic. One, non-orientable self-referential topologies and biochemistry. In Rappaport 2011, a crucial role was ascribed to the topological changes in embryological development and extended to chemistry and cell biology. The non-orientable Möbius band MB and the Klein-Bottle. From now on, we may not explicit a distinction between the surface and the logic as they are fused. As we shall see next, acting in the cell's membrane adhesion sites for integrins molecules that unify the cell's interior and exterior may be related to the logophysical onset of embryonic wave differentiation by changing the topology of the membrane to non-orientable. These are crucial molecules for the self-organization and self-regulation of the cell through its quantum tensegrity structure that extends outside of the cell to the extracellular matrix, conforming thus an holonomic living matrix in which quantum topological superposition and holography are at the roots of cell biology. The living matrix, cytoskeleton, membrane, extracellular matrix extending through the connective tissues to the body, sustaining its integration, appears to act as a perceptual self-transforming system in relation with the environment. Topological transformations of molecules and the cell's living matrix are conceptually similar to the allosteric effect, which is regulatory and is transmitted over a distance, alike entanglement, either topological or quantum, which in the Klein-Bottle logic are related. Within a protein, by which the binding of an effect in one site will change the catalytic behavior of an enzyme or the binding affinity of a binding protein in a different part of the biomolecule. Conformational changes, for example, in enzyme substrate, antibody antigen, protein complexes of protein to DNA are essential to the mutual recognition of biomolecules. These adaptable stereochemical modifications point out to the biological importance of topological changes. This is analogous to the link existing between the topological representation of a given network and the dynamics of the network itself that are supported with its topology, similarly to the dynamical tensegrity structure on which the quantum wave propagation produces embryological development. The classical example is hemoglobin, where the binding of oxygen to one of the 4 subunits arranged at the vertices of a tetrahedron around an axis of 2-fold symmetry increases the affinity of the others. Remarkably, hemoglobin undergoes a crucial topological transformation that allows it to phagocyte. Thus, outside is turned inside, as is the case of the living matrix and the Klein-Bottle, and embryological development, for which also turning inside outside is the case. Thus, the Möbius band MB and KB molecular topologies, due to their topological superpositions and through a quantum field, particularly nilpotent logical operators, can explain the origin of these distant correlations by establishing a logophysical recognition. As is already the case of enzymes in the key and lock model proposed first by Emil Fischer, in which there is a perfect conformational complementarity, the matching is not an artifact of the parts, the emergentist cliché, but the Klein-Bottle logic holonomy over their interaction. A dynamical modification, the induced fit IF to the lock and key model, appeared in Koshland 58, Since enzymes are flexible structures, the active site is continually reshaped by interactions with the substrate as the substrate interacts with the enzyme, alike the behavior of the cell's membrane with respect to the cytoskeleton and their extracellular matrix, as discussed in our accompanying article to the present one. Indeed, while the IF, induced fit, was based upon geometrical and topological related issues, the discovery of a natively unfolded proteins, i.e. protein systems, that perform their physiological role without the need of acquiring a well-defined 3D structure, led to enlarge the idea of structure from a specific geometrical pattern to a dynamical, non-unique configuration. Yet the discovery of natively unfolded proteins, protein systems that perform their physiological role without the need of acquiring a well-defined 3D structure, pushed the scientist to enlarge the general idea of structure from a specific geometrical pattern to a dynamical system moving around a quasi-attractor constituted by a not unique configuration. Thus, we suggest that the determination of the final fit is produced in terms of the elastodynamics quantum tensegrity, which determines the overall dynamics and topology of the cell. In this principle, it's crucial that molecules in an environment can transform its topologies to non-orientable even without breaking chemical bonds. We note that the conventional take on protein structure was built upon X-rays, crystallography, alike the origin of the double helix of genetics. Yet it was remarked by Rich that a method to access the topology already of DNA it does not have the necessary degree of resolution, and through which the double helix was claimed to have been resolved in 2 cases, yet showing considerable stacking. Also, generally, the experimental verification of protein structures is impeded by the actual observation, a feature that also is common to molecules at large. Indeed, the very observation may change the conformation, which is sensible to electromagnetic sources, which are in fact determined already by their interaction with them. We propose that electromagnetic resonance is at the basis of distant communication, and the guidance is informed by the quantum potential, which is also related to the torsion quantum geometry. In the holistic conception of causal quantum mechanics by Bohm and finding final shaping proceeds by topological transformations that may involve an, uh, obliteration of the dualistic separation between exterior and interior. In fact, as was proposed by Boyens, that only a really quantum mechanical theory of molecules and chemical reactions can be realized in terms of the quantum potential, in which the environment plays a crucial role in actually shaping molecules by the absorption or emission of electromagnetic radiation from and to the environment. In fact, as was proposed by Boyens, that only a really quantum mechanical theory of molecules and chemical reactions can be realized in terms of the quantum potential, in which the environment plays a crucial role in actually shaping molecules by the absorption or emission of electromagnetic radiation from and to the environment. Thus, in this absorption and emission of electromagnetic quanta, that molecules interact with the environment and interact between themselves, and shape determinations occur, and in particular topological transformation changing the orientability of molecules. Orientable topologies clearly indicate an outside-inside divide, while non-orientability means that they are intertransformed, as in the case of the Klein bottle. Nej, det är dumhet. Thus, in this absorption and emission of electromagnetic quanta, that molecules interact with the environment and interact between themselves, and shapes determination occurs, as I mentioned before, and in particular topological transformations, changing the orientability of molecules. Orientable topologies clearly indicate an outside-inside divide, while the non-orientability means that they are intertransformed, as in the case of the Klein bottle. The latter is the case of embryological determination. We shall see below that the genetic code has both of topologies. Needless to say, this conception diverges from the present take on chemical reactions, in which no holistics are considered, the environment being neglected. The usual theory, that is, quantum chemistry, appears not to be based on quantum mechanics. And it does not incorporate the environment, rather thinking of chemical reactions as occurring in closed systems. Furthermore, scalar energy landscapes are not the case in chemical reactions, although the usual take assumes them to be the most basic mechanism by vector gradients of the imaginary component of the wave function are crucial instead. This absorption or emission of quanta acts on the wave function of the factorized elements of the overall quantum function, intervening on the chemical reaction, which thus become self-signified, producing a change in the imaginary components of the wave functions, which play the role of the coefficients of time operators. In particular, this is the case of chemical catalytic cycles that define the shaping of molecules by the principle of quenching of angular momentum. This angular momentum is derived from the imaginary phase of the Schrödinger function, and by the quenching mechanisms of the angular momentum determines the stereochemical shape. In the ground state, where the phase imaginary component of the wave function is null, no such phenomenon exists, so that, as expressed before, excited states by quanta absorption or emission, which do have an imaginary phase and thus have an angular momentum gradient, are the basis for this shape determination. Of primary relevance is the shape of the electric charge density of the molecule, which does not arise from the usual hybridization schemes that purport to produce the geometry of atoms and molecules. This density can invaginate due to the topological transformations that occur due to the molecular recognition and that also has a correlate as an elastodynamical transformation whose wavefront is identical to those of the quantum substratum as a light tensegrity structure. Thus, this quantum dynamics does also manifest as an elastodynamics, the latter being the physical field, which is the usual take, is associated to allosterics. I think there's a lot of people wondering what are allosterics. I think they are not related to stellas or sterics. Allo means all. So to resume, these adaptable stereochemical modifications point out to the importance of topology, yet associated to a torsion geometry, both defined by the wave function and secondly, and most important, that these topologies may be changing themselves. The non-orientable Möbius strip and Kleibart topologies of stereochemistry has been claimed to yield a new paradigm to organic chemistry. It was argued it plays a core role in cell biology and embryology, having them all the same organizing principle. In this new paradigm, we contested the dualistic approach implicit to the rigid categorization of cell biology and the phenotypical transformations as linked to an outside and inside ontology, which permeates biology. Indeed, the Klein bottle has no fixed separations between inside and environment due to its self-containment. As a result, the substrate does not simply bind to a rigid active site. The amino acid side chains which make up the active site are molded into the precise positions that enable the enzyme to perform its catalytic function. In some cases, such as glycosidiasis, the substrate molecules also changes shape slightly as it enters the active site. The active site continues to change until the substrate is completely bound, at which point a final shape and charge is determined. Induced fit may enhance the fidelity of molecular recognition in the presence of competition and noise via the conformational proofreading mechanism. This robustness despite of noise is also remarkable about DNA, and embryological differentiation is the very nature of the Klein bottle, since in its informational Hadamard matrix representation, one of its main properties is the fact that it is used as a basis for digital communications due to its robustness. This robustness to the influence of noise is apparent also in the Klein-Bottle topographic map of the sensorium in the human body and the visual recognition of arbitrary patterns that show an intrinsic universal gestalt, which is the Klein-Bottle. We notice the striking similarity with the modification of the living matrix with regards to the environment, which we interpret in terms of the Klein-Bottle logic. We earlier contested the dualistic approach implicit to the rigid categorization of cell biology and phenotypical transformations derived from an outside-inside dual logic, which hemoglobin phagocytosis disproves, yet is pervasive to biology, even in the allegedly radical revisions based on classical physics. Indeed, the Klein-Bottle has no separation between inside and environment due to its self-containment. As a result, the substrate does not simply bind to a rigid active site. The amino acid side chains which make up the active site are molded into the precise positions that enable the enzyme to perform its catalytic function, evidencing a design-oriented change intrinsic to the present logophysical paradigm. IF may enhance the fidelity of molecular recognition in the presence of competition and noise via the conformational proofreading mechanism. Yet, what we interpret as noise might be interpreted as a structured signaling in a nested hierarchy of Klein-Bottles. The hyper-Klein-Bottle. This applies as well to evolution. The topological embodiment of this coupling of an object to the environment is difficult to identify, since, as already explained, the integrity of these entities under study may change their structure beyond recognition. The same problem is encountered with molecules. Yet, catenoid deoxyribonucleic acids in the mitochondria of several cells are pervasive to organic chemistry and DNA. A mechanism for the biogenesis of catenoid DNA on the basis of the Möbius band and for the construction of mBDNA was proposed by Burdick 71. If a Möbius band twisted n times is cut along the midline, catenanes are formed whenever eta is an even number, even when eta is a unity. A single ring is obtained with a double circumference. Other odd values of eta yields various knots. The replication of mBDNA, which is equivalent to this slitting, naturally yields catenoid and knotted macromolecules. Topological operations on DNA are the core of cutting-edge synthetic nanobiology technologies and in the metamaterials. Through topological and self-assembling operations upon DNA material, catenanes, rotexanes, and knots are produced, in which tensegrity structures having multivalid logic enantiomer configurations have a central role. Hereon, we shall deal with the topologies and informatics of DNA in the setting of topological stereochemistry and the Klein bottle. The Klein bottle logic, topological stereochemistry of the genetic code, and informatics. We shall retake the Klein bottle as presented to construct the genetic code. We depart from a logical numerical representation of the Klein bottle logic in figure number 1, in which we have identified 4 states by assigning 0 with outside, 1 with inside, and so that the states are outside, outside, which we write as 0, 0, inside, inside, 1, 1, and 2 transitional states arising from self-penetration, outside, inside, 0, 1, and inside, outside. This does not conform to a dual logic. Rather, it's changing the side of the boundary of self-penetration. Hence, 0 equals 1 and 1 equals 0, as if self-penetration would not be the origin of the boundary. That is the Aristotelian Boolean logic. Thus, we have mapped the topological states of the Klein bottle into a 4-state De Morgan algebra, which is not trivial, since outside 0 is different from inside 1. The latter is, of course, the fundamental metacognitive assumption of science. This is a new representation for the Klein bottle logic to the one derived in 2011 by the author, from which matrix logics that has quantum fuzzy and Boolean logic as subcases is derived. We notice that I and J are the imaginary time waves. that appeared as imaginary local logical values in the calculus of distinctions. We here thus see explicitly their association with the Klein-Bottle self-penetration. They are crucial to the embryology. In topological phenomenological philosophy, free states were considered: the container, outside, that no longer corresponds to the Cartesian exterior space, where objects are passively contained as inside, i.e. the contained, which is the usual take on space, and the uncontained realizing the depth dimension of self-penetration associated to time. An identical free state logic was provided, in which there is a single re-entrance of the form on itself, the archetypical Ouroboros. Yet the distinction between the two states of the self-penetration, transiting between outside and inside, according to which is the departing states, renders the direction of the self-penetration a necessary distinction by itself accounted by I and J. We relate this four-state logic to the four letters A and T. We follow a combinatoric algebraic approach by considering the 2 times 2 matrix label. We shall introduce another distinctions that will be crucial to the topological theory of the GC. Which is the genetic codification. We know from earlier that the invocation of a distinction is tantamount to invoke through the self-entrance of a form produced by this distinction as a boundary cleavage, that is, a Klein-Bottle. In fact, as we shall be considering, 3 distinctions bring into manifestation a hyper-Klein-Bottle. They are produced by three sub-alphabets of the genetic codification, introduced in an algebraic combinatorial framework for the genetic codification in terms of paths, of attributes, anti-attributes, describing succinctly in figure 2 below as depicted, and by the following sub-alphabets. Sub-alphabet number 1, 0 will code for pyrimidines, 1 will code for non-pyrimidines, that is purines. Sub-alphabet 2, amino-mutating or non-amino-mutating under the action of nitrous acid. The same division is given by the attributes keto or amino, so that 0 stands for a letter with an amino mutating property. Subalphabet number 3. 0, a letter with 3 hydrogen bonds. 1, a letter with 2 hydrogen bonds. This is the usual subalphabet. These distinctions introduces further multivaluedness in the topological codification of the Klein-Bottle logic. Yet we shall not tag them with a symbol to distinguish which is the subalphabet they stand for, treating them as binary numbers so that we take 0 outside and 1 inside. Their multivaluedness with respect to the subalphabets will manifest in the Klein-Bottle and the hyper-Klein-Bottle that will appear in the genetic codification. In the sequel, the original interpretation of the matrix elements of P1 by ordered pairs, say C equals 0, 0, outside, outside, will correspond to the concatenation of the first digit corresponding to number 1 and the second digit to number 2. Thus, we have already introduced inside the Klein-Bottle additional Klein-Bottle distinctions, and hyper-Klein-Bottle as nested Klein-Bottles, evidencing a polysemic and polysemantic character of the genetic codification as a hierarchy composed by the Klein-Bottle associated to different subalphabets, indicating the codification of distinct characters. This is illustrated in figure 2 below. Recalling our previous discussions on the coexistence of orientable and non-orientable topologies for molecules, we shall see next that these subalphabets similarly produce the same effects for the genetic codification. In figures 2 and number 3, we go further now, we're looking to the Möbius strip. The line stands for transcription, and the subalphabet by which each operates is the number attached to it. Remarkably, it also presents the symmetries of genome matrices for coding sequences of arbitrary lengths, and their topologies, folding for topological identification according to these three symmetries. Say number 3 yields Möbius band of both chiralities, that is left-turned and right-turned, which followed by either number 2 or number 1 yields the Klein-Bottle. The combination of number 1 with number 2 yields a 2 torus. In the right-hand side of the Möbius band, figure 3, we have drawn the Fischer formula for D-lactic acid, which, if we continue on the surface to the other side, we obtain the L form drawn displaced to the left for allowing its vision. For the genomatrix we have instead the 4 superpositions of either the pairs of opposed rows, which say each first element opposed to the second drawn on the surface, which is quantum interface. For the Mendeleev table, we have instead a superposition of matter-antimatter atom in each side. A continuous transformation changes matter into antimatter and vice versa. The inversion of each element of a pair of rows or columns mandated by taking number 3 plays the role of the antiparallelism in the DH. A simple version of the hypercline bottle with only one Klein bottle recursively iterated instead of 4 is given in figure number 1. And here we have the matryoshkas, and these are 2 possible paradigms of interrelation, hierarchy, systematics. In the LH we find the matryoshkas, recursive containment without self-continuation nor re-entrance of the whole on the parts. This is the current paradigm. In the right duality we have a completely different conception. This is the view of an exemplar of hypercline bottle. The systems re-enter on themselves and may also re-enter in others. The former paradigm is a reduction of the latter by erasing all re-entrances. This is a suppression of the complexity in the etymological sense of falling, so that at first attempt to describe the emergentist paradigm is that it erases that which will later differs to a metallurgical physics that runs by the name of paradigm of complexity. We have thus found 2 fractal hypercline bottle structures in the genomatrix P3 and recursively in Pn1. This goes to show the interpenetration in the matryoshka is very salient when it comes to understanding the different values of inside and outside, but also the interpenetration inside, outside, inside. And this is also very close to the Chattoskotti, which makes possible to have an inside and also to have an outside. So the demarcation becomes possible, and we can see shapes, we can have objects, and we can have dimensionality, something that is quite impossible in the Newtonian approach. So we now go to B-stable perception, and it's perception that changes, which we know is, so to speak, the origin of general perception. How we perceive things is always bistable, it's always double. Monosemic understanding, or the autistic understanding, is that the world is only one. And this is a fallacious way of not being able to perceive the world. It's the lack of perception that gives sustenance to the Newtonian conception, which is not really conception, it's rather the lack of the conception. It's a world without borders, definitions, depth, and so on. The Klein bottle, bistable perception, the double helix, and the non-dual basis of the dual conception of physics and the genome. Having produced the 64 codons at the first stage of recursion by tensor self-multiplication, we proceed to examine the relations with the code of nature proposed for the genetic codification in the setting of NUCRS. This theory is based on recursivity, that is, the algorithmic self-reference establishes a symbolic relation between the Dirac algebra of quantum mechanics and the genetic codification, which is further embodied in a fractal structure produced in terms of the Platonic solids, PS. Thus, by following the construction of the CN and its association with the Dirac algebra, we may establish an isomorphic representation between the Klein bottle logic construction and the CN code of nature. The latter's construction is produced recursively starting from the tetrahedron. At each of its vertices, the four letters A, C, G, and T are positioned, centered in the latter for distinction, similarly to the sign distinction for it in the Hadamard representation above. This single tetrahedron is taken to represent single-stranded DNA, from which, through a fractal recursive construction, the genetic codification follows in terms of fractal concatenations of tetrahedrons. Consequently, all the 64 triplet codons, which are uniquely represented by the elements of the Dirac algebra, can be positioned in the four triangle faces of the tetrahedron, which thus in principle contains the basis for the operation of the genetic codification defined by DNA and RNA. Whatever its topology and geometry may be. We recall that the Klein-Bartel generation of the genetic codification has placed in evidence that the transcription does not require the DH, since the two hyperklein-Bartels found in P3 fully represents the transcription for triplets and further for n-plets by P with n in parentheses. A usually unacknowledged property of the PS drawn in 2D projections centered in one of the two vertices is that the perception gives rise to the so-called bistable perception. That is, two possible percepts connected in time arise depending on the interpretation provided by the other sides acting as cues of the center being in the hind or the fore. Thus, time and depth as the primeval dimension produces this double being coexisting in time. B. This projection was devised in organic chemistry to Emil Fischer to represent enantiomers in 2D and in topological stereochemistry is associated to the Möbius band. Perception is the issue at stake with regards to 2D representation of the stereochemistry of organic chemistry, which produce a multivaluedness that a set of rules for disambiguation is convened to remove them. Herewith, the two possible percepts that arise from the single DNA strand tetrahedron are to be rendered distinct, which the NUCRS, to represent the assumed DH transcription operation of the genetic codification, chooses to represent a star tetrahedron, and thus starting the construction of the CN and the codes of nature and the transcription in the genetic codification. This is the duplication of 3D to the NUCRS and to the codes of nature Bauplan of the genetic codification, and entails a duplication of the Klein-Bartel, which becomes now a hyperklein-Bartel with two nested superposed Klein-Bartels, each associated to one of the tetrahedrons. This duplication has a common singularity by which these Klein-Bartels re-enter the origin. The paradoxical perceptual effect of this duplication is the frustration of the multivaluedness as shown in figures number 6 depicted below, and the duality of the NUCRS and a fortiori the DH and the codes of natures are produced. The code of nature's algorithm starts by using a single tetrahedron in which the 4 bases are placed in each corner. Now the tetrahedron can now be considered to contain in an information level all the 64 possible triplet codons built before from the Klein-Bottle logic. By codifying the 4 bases as in the table number 1 and further taking its threefold tensor products. Well, we go to the conclusions and extensions. We have presented a proposal for the allosteric effect in terms of the same principle for embryological development and still for the membrane and cell biology and stereochemistry. In which shape determination is related to topological transformations that transform an orientable topology with a well-defined interior and exterior to states, to non-orientable in which the logic is no longer dual but the multi-state Klein-Bottle logic. So the Klein-Bottle logic takes its start from topology, space itself, which should be the starting point for any logic. Thus, the fusion of logic and physics, which we have called logophysics, which has quantum geometry as the physical correlate associated to light torsion quantum geometry, has been found to lie as a possible foundation common to biology, chemistry, and also physics, the latter in account of the fundamental role of quanta and light. A theory for the genetic codification has been proposed, departing from the Klein-Bottle logic and its further multi-valued representations of the stereobiochemical structures of the genetic codification to yield 2 hyper-Klein-Bottle and 2 torus representations which do not require the DH. This suggests that need of reconsideration of transcription process. This polytopological character of the biochemistry already appears in the conformational geometry of organic chemistry, specifically in aromatics, and has claimed to be a new paradigm for stereochemistry. In particular, we have suggested that allosterics may also be related to hypotopological accommodation and quantum resonances, already apparent in hemoglobin phagocytosis. Here the role of a quantum guidance, which has as a correlate as an elastodynamics, is the case. The two representations of the genetic codification in terms of hypercline bottles and their biochemical alphabets can be thought as establishing a relation between the genetic codification and the environment in which the latter is transformed into the genetic codification, and vice versa, since the Klein bottle produces this transformation in the very first place. In other hand, the torus structure and its biochemical alphabet of the genetic codification points out to a different way of trading between CG and the environment, in which, so to speak, the CG is isolated within establishing a dual boundary with respect to the environment. In a metacognitive level, this points out to a higher order hypercline bottle, in which both modes are possible. This metacognitive level speaks out by metaphorization, calling for what may appear in first glance as an unwarranted detour. Let us discuss a characterization of metaphorization in terms of a hypercline bottle logic, a family of interpenetrating Klein bottles. Indeed, to metaphorize requires that the subject establish a comparison between A, we call it the metaphoron, and its alleged substitute B. In doing this, there is an identity transparency of the subject with the elements of the relation, and the relation itself is the case of this identity. So we are therefore in presence of three Klein bottles: the subject, the elements of the relation of the metaphor, and the metaphor, and still a fourth one, the metaphorization, as a possibility that enacts the three previous elements. If this metaphor is to be found valid, then other subjects will find meaning and sense in the metaphor, that is, in the relation established by the subject, which has become partially identified with the metaphor and its elements. This commonness of logical value is the logical state of a Klein bottle that contains all the subjects and the elements of the metaphor, and the relation itself between the elements So it is in fact a Klein bottle interpenetrated by other Klein bottles, and hyper Klein bottle as shown in figure 4, as depicted before. Well, that's a variety of the Matryoshka, isn't it? Some subjects will not necessarily assent to the meaning of the metaphor, may place it in doubt, suspending judgment, or reject it in the first place. We all know that the modes of sense of humor are culturally bounded, while a sense of humor is a personal, ethnic or social trait, and all of them together. So here we have another logical values associated to the Klein bottle that contains these subjects, the metaphor and the elements of the relation. This corresponds to the hyper Klein bottle. Anthropomorphinizations are similar, if not identical, to metaphors. As in metaphors, subjects will establish a relation between an element of certain phenomenon and will describe it in terms of another phenomenon which correspond to their domain of action as human beings. Neuroscientists and nowadays physicists, enthused with the notion that the brain is the new frontier of the scientific knowledge, claim that brains think, which of course is not the case. And man-woman thinks. No one has been able to isolate a living brain, and certainly less to find the attributes of the whole thinking person in this impossible to isolate brain. This is an instance of the mereological fallacy that reduces the whole to a part, yet obliterating the whole and its projection to the part, as well as the wholeness of the part. So this metaphor is about self-reference. It is centered in the thinking person with its embodied thinking and thought body which cannot be separated, as in the case of the subject that establishes a metaphor, and by establishing the metaphor, the elements of it have been transferred to the human existence, and in particular its truth value assessment. The metaphor is not independent of self-reference, which is prior to it, be that of the single subject and his fellows that find value in the metaphor. So is the case of scientists that will metaphorize a certain scientific domain, as if it would be universal. The latter we'll call it with the same name that reveals its self-referential origin, the anthropic principle. We have another example here in parentheses from Rappaport. Another such metaphor is natural selection, where the self is the universe in an undetermined claim to be biological sense. Yet to transfer this to claim that the state of the universe is determined by a certain metaphor, say a fine-tuning of universal constants, so that the universe and we exist simultaneously because we pose the metaphor in the first place, is somewhat contorted and reductive way of expressing nothing more than a hypercline bottle in which the commonness of the metaphor is found valid. Yet, which requires in the first place the universality of the family of Klein bottles that make the hypercline bottle and the interrelations, and the metacognitive action of metaphorizing even prior, yet not independent of the others. While they certainly establish a relation between macrocosmos and microcosmos and all the in-betweens, yet not of the trivial embeddings of the Matrushka kind, but with self-re-enterings of an hypercline bottle, as in figure number 4, as depicted. Other different fine-tunings might also be the case for those who cannot find any meaning to the metaphor, just because their language is not the same as ours, and thus communication, if not in the terms of universals, is not possible. These universals return us again, thus, to the hypercline bottle. The fine-tuning stand for the universals of some of the elements of the hypercline bottle, but seems impossible to state that they are valid for all elements of the heterarchy. This would require the claimants to be in a state of identity transparency with the whole heterarchy of the elements of the hypercline bottle, and this is impossible to verify to be the case. Being the latter the case, then we are left with having to establish back to the Klein bottle logic and its ontoepistemology as a first approximation, or still to take a Klein bottle containing an undetermined number of related Klein bottles to elucidate the world in which content and context are unified. This is already our human condition and the condition of all systems. Thus the universals cannot be other than the than context dependent, and this is what has lately been discovered. Physical constants may turn out to be context dependent. To conclude, the hypercline bottle, which we have found to be the case of the genetic codification, and yet is the case of all systems, since this is what is the form of the world, systems are interrelated in hierarchies with reentrances of all elements of them, and other cells and the whole re-entering in elements as well, sustains a world in which for a certain subject of the hierarchy the world seems to be with an outside determined by this subject, and yet re-entrance is maintained to be the case. Consistently with this, say other inhabitants living in another area of the universe will find themselves another fine-tuning to their self-centeredness, and could verify instead another context dependence in their own neighborhood. For an understanding of anthropomorphisms in terms of the differential ontology and epistemology based in the category of distinctions, we refer to Johnson 2008. The detour above showed us that the metacognition implicit in the polytopological character of the genetic codification shows a very rich phenomenology of contextualization. Very rich phenomenology of contextualizations, which is the case of the world at large and of all systems. It is through that rich contextualizations that that only two amino acids as relators of this process of embryological differentiation that appear from the self-referential logophysics in joint action with the environment acting through the remaining amino acids. Evolution, or better said unfolding, with regards to the genetic codification appears as driven by the interaction with the remaining triplets and of the environment at large in a process of contextualization, not as dual two states. Also, the Klein bottle logic provides for the robustness to the environmental action. It further generates a representation for consciousness and its fusion to the physical world. We have re-encountered this topology of the genetic codification and discussed the derivation of NUCRS in terms of two Klein bottles, and seen that space appears to be a materialization of the time depth variable, which is essential to the hemilateral synchronization of the body. Thus, the double 3D structure of physics, one such space being the spatial coordinates, and the other one being the momentum coordinates, has been generated. Since mass is the parameter for momentum space in NUCRS, this has shown that the depth variable that in and through time embodies the B-stable perception of the Klein bottle and generates the double 3D, as we see that time materializes as space. This is another materialization of the imaginary as reality. Indeed, earlier has been shown that embryological development of a Bauplan is such a materialization. The dualistic origin of NUCRS, which requires a duplication of 3D, was found in the duplication of the Klein bottle to obtain a hyper Klein bottle, which is used to represent the DH and the NUCRS, transforming time as an operator on 2D space into a double 3D space, as appears in the NUCRS, one for the physical space itself and the other one for charge. Similarly to the fact that Boolean logic and dualism at large is far from having an independent ontological basis other than the Klein bottle logic, it appears that the DH has the same status, which is quite incredible. We have proposed that this doubling of dimension 3, produced by a superposition of the two Klein bottles, and further suggested in Rappaport 2011 that it lies at the basis of the generation of Bauplans, either physical, chemical, biological, etc., in which the imaginary 3D is related to the time waves in the total generation of Bauplans as a zero-time physical algorithmic causality surmised by Johansen. Its manifestation in the real physical 3D space is a world in which the rigid categorization that separates outside and inside is no longer the case, as shown in the blattospore invagination and in embryological differentiation. In the case of the two blattospore invagination, we saw that it occurs mediated by pairs of Klein bottles as the ovum's aversion takes place, as if the ovum would be thus creating for itself the representation not just of duality but of the mutual interpretation of the imaginary and real 3D spaces. This is more than a plausible observation, but the actualization of the surmountal of the Cartesian cut through the unfolding of what will become a full developed embryo. As shown in NUCRS, This duplication produces the most efficient way for physical symmetries coding and shows up in the rotational generation of the natural numbers from a primeval matrix, which allows to identify the primes completely. Remarkably, the KB structure, the Klein-Bottle structure of the periodic table of elements, in which each atom and stable nuclide of the table appears as if existing twice in each side of the one-sided topology, has promoted the interpretation that there are atoms of atoms and of antimatter. Very perspicaciously, this author has seen that there is a continuity between these two kinds of atoms, which in fact can be transformed one into another by simply moving inside out or outside inwards in the Klein-Bottle representation of the periodic table. We see here the absolute foundation for having any distinctions, to having inside and outside, having measurements, distances, depth, dimensionality, lies in the interpenetration. If there is no interpenetration, there can be no depth. And we cannot have any time, we cannot have any now, we cannot have any past, we cannot have any future. And this is the absolute limitation on the Newtonian take. It's another way of seeing, if you have the Cartesian cut, you will lose out on all ends. Newtonian physics doesn't make it possible to understand the world. It's a way of non-understanding, closing off all understanding, blocking it. I often think of that as a limitation, an obscuring or a darkening. It's very interesting here also to make a comparison to, for instance, the difference between how it's normally in the hierarchy between one would say in a company situation, between the ones that are higher up and the ones that are lower down, where usually the case is the one living lower down are more diminished and not as guided by simple or profound thinking. they're not able to strand into the directionality of a consistent thinking, and in their minds also lies a lower development of understanding. This is the blocking out. What they are working in and what they think usually is a logic is nothing but a diminution and marginalization of logic. This is when it's not enough to think about. So all thinking is always parallel, all thinking is interpenetrational, because it's the very interpenetration that makes it possible to have thinking, to have an understanding. And I think we end there, and I wish you all a very pleasant night, because night